All right, welcome to Bio 388 at Gustavus, where we are shut down. So most of the lectures and most of the material is going to come in this format now. Hopefully, given my background in it, this won't be too big of an issue. We'll still be able to draw on stuff and do my best. These tools aren't 100%, but I feel as though we'll still be able to get this material good. And anytime we do meet as a class, we'll be able to have some questions. I'll probably have you bring in a couple questions on things. We'll be able to discuss some of the good, some of the cool genetic implications of all this, and we'll kind of go from there. So we started this on Friday, and I can't believe that was our last lecture. And we're going to talk, chapter 9 is mainly about chromosomes, but it goes into much greater detail than just, your typical pictures like you're going to see here that you're used to from Bio 101 and things like that. So in this case, we're going to be able to go in and see how DNA can fold up in this like dense, structure right here. For in this case, it's about to get, ripped apart by cell division. That way, we're gonna go that way. Okay. So let's break down these two. So telomeres, when you are replicating DNA, it's gonna grow in this direction, for example. But in a lot of cases, you have those little, remember the little Okazaki, not again. Sorry. Okay, so you have those remember little Okazaki fragments that you have that are going to grow back and forth. So at the telomeres, that's where, if you have an Okazaki fragment, they're going to leave a little bit off every time that they can't replicate. See how there's kind of a gap right here? That DNA then is going to go down at the telomere. So your chromosomes are slowly inching this way, basically dying. They're basically aging. Now remember how this is good against cancer, because when you overgrow tumors, they will just chew ends of chromosomes in tumor cells like you see as moles, on your skin. Although they grew really fast, they couldn't keep it up because they chewed away too much of this telomere, sorry. So that's the basic issue, is that with the direction going 5' to 3' for DNA replication, you're going to have gaps at the very end of a linear chromosome that aren't going to do it. So the telomerase enzyme is a big protein that is going to allow us to extend these sections down here basically indefinitely, because in your gametes, you need to have that DNA be fresh all the time. Okay. So, and centromere, obviously super important. This is one of the more structural sections of DNA, but like Jenna's presentation showed us, these regions do have active genes in them. So that is very interesting. And people had long said this is just, this is junk DNA, this is structural DNA in here, but it plays a super big role. So these sequences and all the 3D topology, as we call it, the big supercoiling structures that exist here, that's what these kinesins are going to grab to and rip apart the DNA when it comes time during cell division. So these are really important structures. And if their 3D topology gets screwed up and their supercoiling is bad, you start pulling things both the wrong way, for example. That is a bad deal. Say that this is screwed up over here. Now both of these copies of chromosome are going to go that way. So that's not good. So we want to keep it over here. All right, so let's take a look at telomerase. Let me try and actually load up. There it is. Okay. So what telomerase is going to do here is that in this big gray thing, TERT, that is a protein. But in this protein is an RNA template. This RNA template is pretty indefinite, and it's going to add free nucleotides to the end. What these free nucleotides are going to allow is that the Okazaki fragments that you're building for this, they can extend on these, quote unquote fake nucleotides up until this section maybe here, and then you never lose that little chunk, because as you can tell, you're adding this extra section that you'll never have to lose then. So that's great. And this is going to be a really good move for your gametes. However, a lot of cancer cells will activate this protein so that they can basically replicate indefinitely, immortal cell lines, essentially. This is like what we talked about with the HeLa cells that can just sit in the plate. And remember, I've worked with HeLa cells. I've worked with a lot of human cells that are immortalized. They can sit in the plate with the media and the resources and just live indefinitely. So activating this in cancers is a key step in a lot of tumors. All right, so We are not going to strictly cover viral genomic material strictly, but remember that, viruses can have a genome that is pure RNA. That's like COVID, has a big protein subunit. All bacteria, though, are going to have a big circular chromosome. And as you can tell, this one has spilled out. We talked about the antibiotic uses against these topoisomerases that are going to keep all this DNA supercoiled. So as you can tell in the bacterial photo, this DNA is completely relaxed. Or, well, I shouldn't use the word relaxed. It is completely uncoiled. DNA does not like to exist like this naturally, but we gave this bacteria, in this case, an inhibitor of topoisomerases or a booster of one or two. You'll see that there are different combinations that unwound the entire DNA genome. And you can see how big it actually is when it is not supercoiled very well. Okay. So a big region in our genome that we're not going to directly super address in this lecture as much because we want to focus on the nuclear genome. Remember that our mitochondrial genomes are these big circular chromosomes. You can call, well, you don't want to call them chromosomes, sorry. I would call them a genome. Big circular genomes that contain all the genes for all the ATP synthases, all the NAD transporters, everything that you need to make this mitochondria work. Those genes are usually in here. They do have some help from our nuclear genome, as this will sometime make products that will come out into our genome a little better, if I can draw an arrow. But for the most part, everything that we're going to look at today is going to, just take a look at DNA pretty broadly. Okay, so this slide is in green. I actually maybe should have put this in blue because this is actually a very good slide for the most part. So what we're going to ultimately cover. So we're going to be looking at DNA unwinding, like in this picture, and you need to do that for replication. Topoisomerases are things that are going to relax the coils or make them tighter, for example. And there's a big interplay between different topoisomerases and like how much that they adjust each other to make sure that this interaction here is going to be good. Why don't I just make a little arrow here? So if this fork right here is not facilitated by a topoisomerase, let's say that we are going to inhibit it. If you can't unwind the DNA, the cell is going to assume that it still needs to replicate its DNA. Problem is that if it's not going to wind very well, then both of these segments are just going to break. That physics tension right there of an underwound DNA, and you're trying to open this for DNA replication to come in, it's just going to snap. Even when there is not a protein facilitating this, let's say you eliminated that topoisomerase, it's just going to break. And that's super bad. These responses are going to try and help the cell. So let's say that this is an antibiotic in this case. These responses will try and help the bacteria cell, but ultimately these are some of our best antibiotics because there's not a really good way to get around this many DNA breaks. Now, a lot of cases, a lot of the resistances, they'll just spit this drug out sometimes. In a lot of cases, bacteria cells will. So we'll get in more of those you can get more into those cellular mechanisms as you study closer, closer principles of medicine in a lot of cases. But these are a great tool to target because if you can't underwind your DNA or overwind it when you need it, everything breaks down. Now we'll see there's one other way that they can get out of this, and we'll save that to the end. Okay. So we kind of talked about this. I should have brought an old phone cord. I know they're just everywhere. And showed you that, this coiling will happen and DNA kind of exists in what we call a relaxed state. It does have, twists and turns. It actually does exist at a certain tension nicely. So when you call it relaxed, this means that you're not under or over coiling it. In this case, with this supercoil, you have to facilitate these coils of coils, right? You have to make sure that this is all, basically the cell has to make sure that it's putting all of these in the right position because to save space, see how much space is saved in this case? That's how much you're going to have to do with your DNA. Your DNA has to be compacted. Now, not only this, it has to be compacted into this state so that it can eventually go and find a nucleosome. So I know we're not on the chalkboard here, but let's call this N right here a nucleosome. The nucleosomes are only going to have DNA wrapped around them as supercoils. Those are supercoils of DNA that are going to be wrapping around those nucleosomes, which remember are histones, and that's remember what we can acetylate and methylate and everything like that. but they have to exist in this big supercoiled structure. This is just too much. There's just too much space. You got to have coils of coils. Okay. So another key thing, remember that antibiotic slide that we just showed, that if you don't have the right topoisomerases or DNA enzymes that are going to, let's say, open or close DNA as it is getting ready all for transcription, then this DNA would just snap. RNA polymerase, as you can tell, is completely separating a DNA strand so that it can make its RNA strand. Now that's perfect, and this is all going to go fine. You do need enzymes that are represented by these arrows to make sure that the DNA is relaxing or tightening back up. If you don't, then this spool of open DNA is going to spill out all over the place. This is all open. These DNA strands will break, and it'll just be a really bad time. Okay. So if you count here, you can count the number of turns here. So let's count a turn here. See, it's going to go here. So there's 1, 2, 3, 4, 5, 6, 7. So it should have started right there. Anyway, there's more. This is a tighter set of turns than this one. Now, this is called strained because you're basically going to strain something if you go to 7, or let's say if you went to 9. So if you hypercoiled this or less coiled this, DNA does not like to get out of this relaxed state. You have to force it into that supercoil state, because otherwise it's going to try and stay at this 8-turn conformation. So anytime that we're doing this, that means we're going to put some pressure on this molecule, and you are likely going to have to use ATP to put that in the right position. Now, D is the most important. As you could have told in the last slide, you need to be able to access DNA. That's the key. RNA polymerase needs access to this area for any gene to turn on. If you lose the topoisomerase ability to get into this DNA, let's say that it's even acetylated, this is the next step. You need to have transcription factors able to just bind this section and then turn on a gene, basically. Let's just move this. Still getting used to the arrows, trust me. But again, this is another gatekeeper, just like acetylation versus methylation of the histones or the DNA itself is. Okay. Now, where supercoiling is super important is at this metaphase and anaphase stage. This is an entire chromosome here. So it's millions and millions of base pairs all crunched down. So the scaffold for the chromosomes there is going to be like a protein structure scaffold that is going to allow binding to certain points. Give me that. There we go. It's going to allow binding of this supercoiled DNA at certain points that's going to keep it in this characteristic chromosome structure above. But as you can tell, this DNA is highly, highly wound, compact. It is twisting around. It's going all over the place. It needs to be in the tightest possible conformation, and a lot of work goes into your cells to put it in this conformation without breaking it. That's where all these topoisomerases and gyrases are going to come in. They need to compact this DNA because, like we saw in the intro slide, oh boy, just draw an arrow, come on. Because okay, never mind. Like we saw in the intro slide, one direction needs to go this way, one sister chromatid needs to go that way, and you need to pull those apart. That means you need all your DNA all set up for this journey, all compact. Oh, that didn't bend as much as I wanted. Okay. So this is a very good time to address gene therapy and sometimes why it fails. So this integration step, this is for retroviral gene therapy. I'm going to just explain that. This figure is going to use an adenovirus. Okay, so this is more like what you see in a COVID. So you have a virus, it's in a big protein capsid, but it has its nuclear material. We have inserted a gene. This is going to be for patients that were born without a certain gene, like they were double recessive for something. So imagine this is the CF, the cystic fibrosis receptor, for example, and we're going to put that into somebody's cell. We're going to target this virus to make sure that these targeting receptors here are going to target, let's say, the lung cells for cystic fibrosis. Perfect. That is going to go, it's going to find this cell, and it's going to inject itself into the nucleus. So let's take a look here at this zoomed picture. Adenoviruses are going to inject their genetic material, and it is going to get made into mRNA. There's no integration at this point. So that's why adenovirus vectors are usually a little safer when it comes to gene therapy. Now, remember integration of the genome? Remember how that is like HIV? A lot of the reason why gene therapies fail because of integration, or when you try to do a retrovirus, because remember this, you're going to have to keep infecting someone with this virus to keep getting this good result. And I mean, remember, this is a theoretically harmless virus, but you're still manipulating a lot of machinery here. The integration of a retrovirus was thought to be, oh, this is good because this will be permanent and we'll just put the gene back in. The problem is that our cells reject that a lot of the time, not only because the immune system will see that coming in and think it's foreign, but the structure and the coiling of the DNA just does not favor integration at the right areas. And it won't integrate in the right areas all the time, for example. And so the topology and like how tight certain regions of DNA are, things like that, it's not going to go in the place that you want it to go sometimes. So this probably should have been a blue slide, just to clarify that. This is obviously a good thing to cover, but maybe not as direct like as this one. So we talked about the importance of the centromeres and how those are going to connect. And the kinetochore microtubules, see these big structure tubules? They're going to pull. But let's say, I do not like that function. Okay, there we go. Now let's say you have this chromosome here, but that this centromere is just not very good for whatever reason. It's got a bad structural topology, or it wasn't wound up enough. Anything could happen. You're going to get the whole thing moving over to this cell. Okay. Now, if this happens in one of your normal cells, yeah, now you have a cell that is missing this chromosome. This cell that divides here is probably going to die. But you do have this cell over here moving. Now it has 2 copies. If this has a section, let's say, now you have extra copies of genes, that may kill the cell, but that may give it growth advantages that can lead to cancer. For example, now instead of 2 growth genes, you have 4 copies of the growth gene, right? And that wouldn't be a good time. Or at least not 4, but 2 with the sister chromatid instead of 1. But you would have 3 total then, because you'd have another chromatid of this one. So when you ruin this section of a chromosome and it is not like nicely wound, and it's not going to attract the right kinetochore microtubules to attach to it, then you're going to sometimes just take the entire thing away when you should just be taking 2 away like that. You might get the entire package accidentally. Okay, so we talked about those wild inverted DNA structures. So remember, this is a double cruciform. So remember, that means that this repeat is going to match with this repeat. So we're going to draw that. And then this repeat is then going to match with this repeat. Now, for this wild structure to actually form, you do need to just at least underwind the DNA. You're never going to have hairpins, cruciforms, or anything like that if the DNA is not open, and you're not going to be able to access it like we have here. So there is a whole extra step to these wild structures that you do need to account for, and that is this DNA needs to be open and underwound out of its natural relaxed state, which is about 8 turns. As you can tell, open DNA does not have 8 turns. It has much fewer. Sorry. All right. So we have talked about nucleosomes. Remember that these are made of those little histones. So these are made of 8 histones right here, and they are basically they're like a little octamer. So there's 2 groups of 4 that are stacked on top of each other. So 4 times 2. Oh, this is ugly. And you can form a big histone. Okay, so DNA, as we write it on chalk, I've been writing it like this, and it just goes around, and that's your histone, right? In reality, this is supercoiled DNA that is wrapping around. Okay, so this wraparound DNA here, that is highly compacted DNA. It is highly silenced because of that. The only way to open it is to add, remember, those acetyl groups to your histone. That's going to break the DNA off and allow it to be really open. Oh no. And so that's why it can be so, compacted, is that, this structure right here, see how many turns are in this? It is really crunched. And so anytime you have DNA that should be inaccessible for long periods, or at least, like in a cell that doesn't need that gene on, it's going to stay crunched like this right up to its nucleosome and basically never activate. Because like I said, remember, it's much worse to have a gene activate that shouldn't be activating. All right, LK, this is fun. So this is going to measure basically how, what direction are you tightening or loosening the DNA. So an LK01 means that, yeah, there's not a lot of twist and there's not a lot of turns. So the twist is like kind of this number of, helixes that I'm drawing right now. The writhe is, let me make sure. Nope, never mind, that's writhe. Sorry, see, this is hard. So the writhe is the amount of ups and downs that I just drew. The twist is, see how this is kind of like a very flat ribbon and the whole thing is kind of just facing itself? See how this ribbon is facing in and out and in and out. So it's like a corkscrew. That's twist. And so both of those factors add up to LK. Okay. So this is a good spot to pause right here and just kind of, and again, I know that books aren't everybody's favorite thing, but especially with our format right now, please do look into the details of this, because with how the exam's going to be now, and especially sections like this that are now purely online, the level of detail is going to be there that, I wish could have been there in class. We will have question time for this. So if you literally have questions, now's the time. I'm still going to have open office hours on Wednesday. You can still just, Google Meet. Everything's going to work like that. We'll see how many people are up for that after St. Patty's Day on Tuesday. But goddamn, be safe, please. Like, don't surround yourself with people that could be sick. All right, back to the lecture. That's going to be a hidden gem for anybody that gets this far. You have relaxed DNA, as you can tell up here. This is the natural state. These large writhing turns, even though they are not twisting the DNA and corkscrewing it, they are like much tighter, as you can tell, going up, or well, in this case, much looser it looks like. But a large twist, this is also another way to compact. So when you have twist plus writhe, you can really compact something. So here's the big deal with DNA, is that you can supercoil in both directions, okay? You can kind of tell the difference in these images by looking at them and seeing what direction they've been coiled in. But remember that there are now two forces that can regulate this usual setup of DNA. Most DNA is going to exist in this state if it's about to be manipulated, and you can control it by taking it this direction or this direction. And there are different genes that are going to do different things. So let's cover the first one that is going to cover negative supercoiling, okay? So those genes that do negative supercoiling are going to be fought by genes that do positive supercoiling. So let's meet two of those. All right, this is a gyrase. This is going to put DNA and induce negative supercoils. This will take ATP to do it, but essentially it has certain gates, active sites, that it is going to wind the DNA. So you've seen this as cartoons before. This is the first time that you're going to see this as something fluid, something actually happening, an enzyme actually breaking DNA and winding it tighter. I would highly recommend looking at some kind of animation for this. We'll do our best right here. But again, doing a video of this is a great way to learn it. Okay. So like this says, to twist the DNA, this tyrosine is first going to make contact, and it is going to break this DNA strand. That's how you twist DNA. Now this other side of this gate, see how it's going to swing this around here? It is going to swing that strand in here, and it's going to reanneal the strand that you just cut. And now you've twisted DNA one time, okay? You literally, that's how you relax or induce coils. You break the strand, okay? You are playing with absolute fire here. Every time your topoisomerases are doing this, and they are doing it constantly to relax, unrelax DNA all the time. And there are opposing forces, remember, because we just induced a negative supercoil here, not a positive one, so the other direction can happen. You are breaking the DNA when you do this and passing it through. That's how you make a loop. So you can imagine that issues, let's say with this amino acid section right here, can lead to profound issues in the cell. Topoisomerase mutations are found frequently in cancers that have incredible genomics instability. Because if this interaction is ruined, you are making breaks all the time that you're not fixing properly. That's how you lead to disease. Okay, now positive supercoils. So remember, these are the same type of thing as gyrases, but in this case, it coils this a different direction. So you need an entire new enzyme, and in this case, you do need ATP. So gyrases, we did not use ATP here. This was just facilitated by the tyrosine. Okay, and that's a nature of going negative versus going positive. Okay, again, you are going to have a big topoisomerase. It's going to grab DNA at this stage. Oh, wait, no, this is, we are still in gyrase. Never mind, I'm sorry. Yeah, sorry, this is topoisomerase up here. I'm sorry. Okay, and this is gyrase. Sorry, what a disaster. Okay, so G for gyrase. Okay. Oh my God, give me a break. Okay. So this slide is G for gyrase. I don't know, that doesn't look like a G. Gyrases are going to use ATP again, so they are going to break the strand. So see how they bring the strand together here at this gate right here. It's going to crack open one of these strands, and it's going to pass it right over the other. And now you've induced another negative loop. So you've induced a negative supercoil here. And so you call these little loops that you make each time nodes. But look at this thing is just going to keep making node after node after node. And it's going to keep winding this thing over and over again. So remember that we talked about the antibiotics that can kill bacterial topoisomerases and leave human ones unharmed for the most part, right? The way that they develop resistance is that when you lose one of your positive supercoilers, let's say, so a topoisomerase, all those bacteria are going to go down if they don't figure something out soon. That's extinction, okay? Now what happens is that you have taken away a positive supercoiler, but that means that a mutation in one of your negative supercoilers, if you lose that negative supercoiling, then this positive supercoiling that you lost has been compensated for. These are called compensatory mutations. Basically, you're inhibiting a plus like coiler, and so if you're going to eliminate that, the bacteria that resist will have their negative supercoilers. They'll eliminate the negative supercoiler that usually is going to work against the positive supercoiler. Yeah. That you were inhibiting. So most gyrases and topoisomerases are working against each other in kind of a competition that ultimately leads to a healthy genome. So if you lose the positive supercoiler, you're in really bad trouble because you have imbalance. But if you get another mutation in one of the opposing ones, then you can go right back to a really high fitness. Now sometimes, so over here, God, this thing stinks. Okay. So in this case, sometimes those mutations just take you back to the fitness that you were at, but other times they take you to an even more resistant level because you've made a new strain of bacteria that can never again be touched on its topoisomerases for this, for this antibiotic in that case. Okay. And again, remember, so to kind of illustrate the point that topoisomerases, remember, you're breaking these strands. So even chromosomes that are, together like this, when you unwind them, you will literally be breaking the strands apart, and everything is going to come nice and nice and free. So this is a nucleosome. Consider this the histones right here. To get them around these histones, you do have to negatively supercoil them. So these enzymes will have to wind the strand not only around the DNA binding protein, but around themselves so that you have, some good structure here. Okay. There are also topoisomerases that can go both directions, as you can tell. So these are big multi-unit proteins. One is going to be grabbing one section of DNA. One is going to be grabbing another double strand of DNA, and they can grab both. They will cleave this, and the orange right here is going to be marked in your book. They're going to cleave this, and this orange section is going to pass right through. Oh boy. The orange section can now pass through this break here, It's going to pass right through here, and now it's over. So that kind of illustrates what happened in this case here. These had to be broken to get relieved. So during the compaction of DNA, during cell division, you're going to have different, basically these SMC proteins right here. These are going to compete to compact versus elongate DNA. Okay? Cohesins and condensins are going to keep DNA compacted. They're going to make sure that they stay in a good spot. And so here's a very good illustration, starting here in G1. You have a massive chromosome. These cohesin proteins will start to wrap around the chromosome, then it replicates in S phase, and they're going to keep all this condense as it's replicating, except for the areas that are, physically replicating at that time. Then condensin shows up. Now condensin is just one of these little topoisomerases right here. It is going to connect DNA and make sure that it stays compact. That's what this little structure is going to do. It's going to bind here, and it's going to bind up all the DNA. So that's what condensin is going to do. It's going to completely supercoil things down to this. So see how small this is now comparatively to how long this chromosome was. And this picture can't even really do it justice to how much space it's saving. Then separase is just a big enzyme that is going to cut these cohesins off. So remember, the cohesins are more structural. They're just keeping the DNA in the right place. Separase is going to cleave all these away. And now the chromosomes can get pulled apart by the kinetochores, which are going to be seizing on those centromeres that we talked about, the structural components. And then they're taken away. And then they are elongated again and allowed to go back to that DNA relaxed state. So it's this big interplay of, if I could draw here, now I'm drawing too much. It's this big interplay of, okay, you are very relaxed, you're very relaxed, now you're a lot tighter, you're still tighter, then you relax again, and you go down. So it's a very regulated, kind of orchestrated process that needs to happen every time you're going to put this DNA through this. So the rosette is how you condense it. This is how you hypercondense something. Remember, this is during metaphase. Okay, this is when chromosomes are so dense that you can actually see the DNA on a microscope. So you start with just elongated chromatin. You have condensin. You have these looping proteins right here. See how they're going to loop? This looping is going to facilitate binding not only between the little proteins, but it's going to start adding lots of twist. Or sorry, this is going to add lots of writhe. See, this is hard. Remember, I'm not going to ask for strict definitions because remember, you're going to have most of this accessible to you. I can't police that. You're going to have these little rings. That's when the magic starts happening, is that during this prophase, these rings are going to interact. They are going to all form very intricate rosette structures by pulling on each other from this stage. See how this, oh, let me do that again. Let me actually delete this too. Okay. See how this section is ready to connect with the purple? It's going to connect with this purple section right here. And it is going to form this immense, awesome rosette structure here. And to this day, the rosette structure is not something that we can 100% confirm, but there is not a likely better scenario for how condensed DNA can get. So you have these massive supercoils and these massive helixes that are going to stack on one another. Then you start stacking rosettes one after one on top of the other. And it's a really neat way that you can store that much DNA. All right, so you may have seen this in the news. Clover Queen. There's a million ways to say this. I'm probably saying it wrong. Clover Queen is something interesting. This drug, this compound is this strange cure-all. I hate using that word, but it seems to have a role in almost any disease you look at. So this stabilizes the underwound structure of DNA. It stabilizes, we don't know why strictly, but we think that it has a role as far as, regulating topoisomerases to make sure that they have that relaxed state. And it's really strange. It also is responsible for bringing more zinc into the cell. It also really inhibits autophagy in cancer cells. So that's when the cancer cells are eating contents of their own cytoplasm for more energy, basically. It inhibits that, so it inhibits cancer cells. It's something that pops up all over the place. And I mean, like Dr. Burr in chemistry uses this, this is how you treat malaria, for example. Like I've taken this. And right now, with COVID out, this is one of the things that's working. And it's theorized because of the zinc factor that it brings in zinc. Literally, it does all these weird things, and none of them are very related to each other as far as we can tell. So it's one of those mystery compounds. We know it works. We don't know why. So as you move this direction, you're more towards that relaxed state. As you can tell here, you have 0.5 micrograms, but if you move to 5 micrograms, you can tell that this shift right here has moved far towards that way more relaxed negative supercoil state. So it induces negative supercoils. Now this is a good thing. I mean, as far as we can tell, when treating the malaria protist for whatever reason, maybe it messes with the malaria genome as it relates to the good amount of negative supercoiling that you want to see. And you'd have to read a little more on its direct effects on those topoisomerases and gyrases that we talked about. But remember, it could be boosting a gyrase to inhibit or to move more negatives, so it could be boosting a gyrase, or it could be inhibiting positive coiling topoisomerases. So remember, whenever you're looking at DNA supercoiling, you have to remember that these are two forces that are opposing each other to twist one way or another. So do your research on CQ, as I've always called it here, because it's pretty much one of the only things that's working on COVID right now. And remember, like I've said, this is a very strange molecule. I wouldn't want to take it if I didn't have to, but that's about it. All right, everybody, stay safe. And like I said, do practice some social distancing if you go out this weekend. Stay healthy and have a good one.